Good afternoon. Great to see everybody out today. Hope everyone's enjoying lunch. I'm going to do an introduction for Dr. Bhatia, and then turn things over to him for his presentation. So this is Dr. Ravi Bhatia. He's a distinguished professor in the Division of Hematology and Oncology here at UAB. He received his hematology and oncology and bone marrow transplant training from the University of Minnesota, and then joined the City of Hope National Medical Center in 1996. Began his career as a physician scientist. Joined UAB in January of 2015 as a professor in the Department of Medicine. And then up until 2021, he was the director of the Division of Hemonc and also deputy director of the O'Neill Comprehensive Cancer Center. Served as interim director of the Comprehensive Cancer Center in 2019 and currently director of the stem cell program here at UAB. His clinical interests are in hematological malignancies, hematopoietic cell transplantation, with emphasis on myeloid leukemias. His research interests are in studying the regulation of normal and malignant stem cell therapeutic targeting of malignant stem cells and hematopoietic stem cell therapeutics. He's authored over 200 publications as a recipient of NIH grant funding continuously since 2006. He's been a scholar of the Leukemia and Lymphoma Society, an elected member of the American Society of Clinical Investigation, and of the Association of American Physicians. Today, Dr. Ravi Bhatia is going to be speaking on the elusive leukemia stem cell.
Thank you very much for the nice introduction. It's great to be here amongst friends, mostly, and and a lot of people who are known for a long time, including some who came back. I'm very grateful. Yeah. And I was looking back, and I think I did pathology grand rounds right about the time I came. That was September of 2015. I think maybe Adam had a role in sort of getting me in at that time. But I'm really pleased to be speaking again and giving an update on some of the work that we've been doing. So my lab works on hematopoiesis, normal and malignant hematopoietic stem cells, studying the mechanisms of regulation, and particularly for leukemia stem cells, the mechanisms of response and resistance to therapy. We've been particularly interested in the role of the bone marrow microenvironment in regulating normal and malignant stem cells, characterization of the bone marrow niches for these cells and regulatory mechanisms, and the interactions with intracellular signaling and intracellular regulatory networks, metabolism, epigenetics, and trying to use this hopefully for developing strategies to eliminate leukemia stem cells, which are hard to eliminate, and enhance possibility of cures for patients. And the other area which we work in, I'm not talking about today, but hopefully sometime in the future, is really understanding therapy-related myeloid neoplasm, therapy-related MDS and AML, and the pathogenesis and prediction of this, looking at early stages of development of this malignancy, and understanding and developing ways to predict patients at risk so that you can do early interventions. So just an introduction to hematopoietic stem cells, with which, I think many of you are, most of you are familiar, but hematopoietic stem cells are, stem cells which are at the apex of hematopoiesis, have the capability of giving rise to all the different hematopoietic lineages, including myeloid and lymphoid, as well as erythrocytic, megakaryocytic. They go through phases of differentiation from HSCs to multipotent progenitors to more progenitors with more limited capacity, and finally to mature cells. And conceptually, we've got, especially with the advent of cell tracing and single cell analysis, we now have a sort of a concept that these aren't really necessarily discrete phases, but a continuum at the apex of which is the stem cell, which is characterized by being quite dormant, quiescent, having extensive self-renewal capacity, having low cell cycle activity, high autophagy, low levels of metabolism and translation, being primarily glycolytic dependent. And then cells getting activated, with increased AXPHOS, increased metabolic activity, and differentiating into multipotent progenitors, then getting lineage priming, and then becoming the different lineage progenitors and mature lineage cells. So the disease I'm going to talk about today is chronic myelogenous leukemia, which, as you know, is a malignancy arising from a hematopoietic stem cell. It results from the 922 translocation, which was, results in the Philadelphia chromosome. The translocation was leads to the formation of the abnormal BCR-ABL fusion oncogene, which has abnormal tyrosine kinase activity and which, when expressed in the stem cells, gives rise to a myeloproliferative disorder because of constitutive activation and abnormal activation of a number of downstream signaling pathways. In spite of the myeloproliferation and everything, the CML, the stem cell that gives rise to CML, is very similar to its normal counterpart. So, just like in normal hematopoiesis, CML also has been shown to arise, from a stem cell, which expresses phenotypic markers, similar to that of normal hematopoietic stem cells, CD34 positive, 90 positive, 38 negative, RA negative, very quiescent, low metabolic activity and translation, more glycolytic dependent. And these quiescent stem cells are present in CML bone marrow. And as we are going to just talk about it, they are quite important. So when I was training in hematology oncology, the treatment of choice for CML was the bone marrow transplantation, and that's a great treatment for CML. It's the one treatment that can known to give rise to long-term disease-free survival. But around the turn of the century, there was the advent of BCR-ABL tyrosine kinase inhibitors. The first one was imatinib, which basically targets the ATP binding pocket of the BCR-ABL kinase and prevents substrate phosphorylation. And these have, as you know, transformed the long-term outcomes of patients with chronic phase CML and lead to deep responses in the majority of patients and to a life expectancy, which is now similar to that of the general population in responding patients. And now there's been evolution. There was some resistance to imatinib, and new TKIs were developed, which were more potent and bound to mutant TKIs. And there are now 6 different TKIs or BCR-ABL inhibitors, which are approved for the treatment of CML at present. One of the things that we found quite early on was that, we were interested to see whether these TKIs were able to target the BCR-ABL positive leukemia stem cell in CML patients. And we did basically simple experiments in which we isolated CD34 positive, 38 negative stem cell populations and then analyzed them for presence of the BCR-ABL gene, both by FISH or by expression. And we could see that there was persistence of BCR-ABL positive stem cells in CML patients, many years out from treatment, up to 6, 7 years out from treatment. And these were patients, who had achieved, deep molecular response. And this has been sort of confirmed by many other groups and is sort of an accepted sort of dogma right now in the field. And, we and others have shown that TKIs, it's not like they don't act on stem cells. They're active on stem cells. They inhibit their proliferation, as shown here. This is a CFSC assay. But then the CFSC bright dormant stem cells, tend to remain present even in the presence of TKI. So it is not effective against the quiescent, as effective against the quiescent leukemia stem cell in terms of eliminating them. I'll just introduce this mouse model because we've used it in a number of our experiments. It's the SCLTTA TRE BCR-ABL transgenic mouse model. It's targeted BCR-ABL expression in murine stem cells via a TET-regulated promoter. So if you withdraw tetracycline, BCR-ABL is expressed in the stem cells, and mice develop a myeloproliferative disorder, kind of like CML with leukocytosis and splenomegaly. It's a little more aggressive than what we'd normally see in patients. But we've shown in this model that the stem cells in this model, the leukemia stem cells, share the same phenotype as normal stem cells, just as we see in patients with CML. And we've done many studies, some of which, I talked about the last time I was here, in which we've tried to understand, what are the mechanisms by which, even after TKIs inhibit BCR-ABL, stem cells are able to persist, especially, I think we and a number of other labs have shown that, there are persistent, epigenetic changes and transcriptional networks which are active in these cells, involving, P53 and MYC and SIRT1, BCL6 and others, and epigenetic changes which are associated with persistence of the stem cell and which, can be targeted to deplete the stem cells, also a role for metabolism and autophagy. And then importantly, that the stem cells, have a close relationship with the microenvironment. Actually, in CML, there are alterations in the microenvironment so that the microenvironment is now quite receptive for the maintenance of leukemia cells. This is a feature that we know from many other malignancies where the cancer sort of adopts the alters the microenvironment to help maintain, the cancer cell. And there's a lot of interest in the role of inflammatory chemokines, cytokines, etc. In maintaining leukemia stem cells. So what is the importance of this? The importance of the fact that leukemia stem cells are not eliminated is that, if you were to stop treatment, in many patients, the majority of patients, the leukemia will recur. And so that means that the majority of patients would need to take continued TKI treatment potentially for the rest of their lives in order to keep their leukemia under control. So that's not necessarily the worst thing. It's much better than we can do for most other cancers. And if people take their drugs and are remaining in remission and have a normal lifespan, I mean, that is fine. But there are issues with this. There are adverse effects and potential intolerance. A lot of them are, things like fatigue and pains and cramps and stuff like that. But there can be serious adverse events as well, including cardiovascular and pulmonary adverse events from some of the more, advanced TKIs. Then there's a strong possibility of lack of adherence with taking a drug every day for your life. And we know that with lack of adherence, even a small lack of adherence can be associated with risk of treatment failure. With this, there's a possibility of acquisition of drug resistance. And then for young women who want to be pregnant, these drugs are teratogenic. So, you have to come off these drugs in order to become pregnant, to have a safe pregnancy. And then these are terribly expensive. They cost, from 150 to 250,000 a year. And if you multiply that by, decades of treatment, then that's a lot of money being spent on these treatments. It's much more than what we'd spend, say, on things which we think are very expensive, like bone marrow transplants and CAR T cells and things like that, if you take the cumulative expense. But the good news is, and I think we always learn from our patients, and I'd say that, in terms of what we're seeing in CML, I think the clinical experience has been, I think, ahead of where we are in terms of understanding the biology, but it's helping us to think about how we are understanding the biology. And what was found was that there is a subset of patients who achieve a deep molecular response, which means that if you do a quantitative PCR, they have a more than a 4 to 4.5 log reduction in BCR-ABL. That these patients, if they can maintain deep molecular response for an extended period of time for more than 2 or 3 years, and have been treated with TKI for 5 or 6 years, then it's possible to stop the TKI, and in about half of these patients, the disease will not recur. The other half of patients will have disease recurrence, as shown here. But the good thing is that in patients in whom the disease recurs, the treatment can be restarted and the patients can go back into remission. And this whole process is very safe as long as you're carefully monitoring the patients after you stop treatment. And so this is very interesting. And, so how does this fit in with the fact that the stem cell is resistant to TKI. And so clearly in some patients, the stem cell can be sufficiently suppressed so as to prevent disease recurrence. And so, how is this happening? And really the question now is, how can we make this happen for a larger number of patients or for all patients with CML? Because if you think about it, with patients starting off on treatment, say even, 40 to 50% of them get a deep molecular response, and half of them are able to maintain a remission, you're still at about 20 to 25% of patients who will be able to achieve a treatment-free response. That still leaves 75% of patients who need, lifelong treatment. So, there's clearly a need, and this is probably the biggest sort of area of importance in terms of CML research right now, besides, a smaller group of patients who tend to be resistant to TKIs, who don't tolerate TKIs, and who do badly because of disease progression. But this is still the bulk of patients. So some people have developed a more sensitive assay, which is a DNA PCR assay, which has a sensitivity down to 6 logs. And because the breakpoint is different in every patient, you have to basically have a patient-specific assay, for that individual breakpoint. But you can get down to a greater degree of sensitivity, down to 1 in 10 to the 6. Regular PCR assays are sensitive down to about 4.5 logs, and this is 1.5 to 2 logs more sensitive. And what you can see is in this study from Australia where they did this PCR, the 0 point is the time of imatinib discontinuation. And after imatinib discontinuation, for up to 10 years after treatment, you can see this persistence of the BCR-ABL positive cells in these patients who are maintaining treatment-free remission. So it means that, a cell that's capable of long-term persistence or long-term generation of BCR-ABL positive cells can still hang around, although the patient remains in remission. And it's interesting too that the levels of residual disease, can actually decrease over time even after stopping treatment. So there's something happening in the patient that even though these BCR-ABL positive cells were there at the time you stopped treatment, they tended to sort of go down in terms of their levels, at least in some patients. So this suggests that in addition to suppression of leukemia stem cells, the lack of recurrence or recurrence versus lack of recurrence may be influenced by heterogeneity of the residual leukemia stem cells, heterogeneity possibly in their quiescence or in their regenerative potential, or host factors, things in like the microenvironmental niche, immunity, metabolites, other things in the microenvironment which influence the ability of the stem cell to regrow and regenerate leukemia. And, this is not unique to CML. These are things which you sort of know of in other solid tumors. It's quite common in prostate cancer to have cells, hang around in the bone for years and years after the prostate is removed without, mets developing. So it's this thing of long-term cancer cell dormancy with possibility of growth is a common problem. So the questions are, what are the characteristics of the stem cell population that persist during molecular remission? And what determines whether they regenerate leukemia or not after stopping TKI? And I think that's kind of what we're hoping to sort of learn more about, as we continue our work now. The challenges are that, these cells are really present in minuscule numbers. So, they're a very small fraction of bone marrow cells. And then if you're trying to compare leukemic versus normal stem cells, they're an even smaller fraction of the stem cells in the bone marrow in patients in remission. So they're hard to identify, isolate, and study. And then there's the issue of heterogeneity, and heterogeneity could be genetic, epigenetic, cellular, could be stochastic, and how do you study that? And then, I think as we study regulatory pathways and regulatory mechanisms, how do you find out which are really the key mechanisms as opposed to, having redundant and interdependent mechanisms? How do you find the nodes that are important in order to, sort of have the effect that you want? And then the other thing is the complexity of the microenvironment, which we'll discuss. And how does the interaction of the microenvironment with leukemic stem cells versus normal stem cells differ? And how do you dissect that? And how do you model that and replicate it and manipulate it? So just a word about hematopoietic stem cell heterogeneity. So hematopoietic stem cells are known to have functional heterogeneity in terms of their lineage bias. So, you think of a stem cell as a cell that can give rise to all the different lineages, myeloid and lymphoid, and that's like the balanced stem cell. But there are stem cells which have characteristics of being more myeloid biased or being more lymphoid biased. So, this is one thing. So it may depend on the type of stem cell which is persisting after treatment. And then there are other causes of stem cell heterogeneity. The localization to the niche may, play a role in determining stem cell heterogeneity. There may be, differences in genetics in terms of additional mutations. There could be epigenetic heterogeneity. It may be biological processes like transcription, metabolism. It could be related to segregation of cell fate determinants, or it could be simply stochastic. So this is one of the challenges. Uh, we know from the work from the same group that did the DNA PCR, they sort of went further and they fractionated different populations from blood of patients who had stopped TKI. And they compared patients who maintained treatment-free remission versus those who relapsed. And they found that the presence of BCR-ABL positive white cells was associated with increased risk of relapse. But also higher numbers of BCR-ABL positive granulocytes and T cells were associated with increased risk of relapse, but not so much B cells and NK cells. And so this might suggest, based on what I showed previously with the different stem cell types, that it's possible that, when you have something like out here, you might have a more lymphoid biased stem cell, whereas out here you may have a more, a stem cell with a different capacity or with greater myeloid biased stem cell or a more, balanced stem cell that may be more capable of causing a relapse. Now, this is a very nice study which was published several years back by Adam Mead from Oxford. And, what he did was this is early single cell analysis. And this is not 10x. This is actually taking individual cells, sorting them, and splitting the DNA and the RNA. And this allowed him to actually look very clearly at BCR-ABL expression as well as gene expression in the CD34 positive cells. And what he found was that in CML, the black is the normal and the blues are the CML stem cells. And in CML, there were like some heterogeneity of stem cells. They were like, he classified these as two groups. And he found that this group A was the group that tended to accumulate with TKI treatment, whereas group B went down. And the group A stem cells had characteristics of quiescence, greater stem cell associated disease expression. They were the minority at diagnosis, they increased, and they were also enriched for these inflammatory gene profiles with TNF alpha, IL-6, as well as TGF beta. So this suggests again that there is a heterogeneous population of stem cells, and there is a particular population which is more TKI resistant, which tends to accumulate over time. Another group from Lund in Sweden has also looked at heterogeneity within leukemia stem cell populations. In CML, this is at diagnosis. And they were able to identify leukemia stem cell subfractions which had primitive and quiescent signatures as opposed to being more activated. And they showed that the residual leukemia stem cells after TKI treatment were enriched for low levels of c-KIT expression and for higher levels of CD26 expression. So this primitive lineage negative stem cells without c-KIT but expressing CD26 seem to represent the more primitive TKI resistant population. And it's interesting because, we studied this using our mouse model, and we sorted CML stem cells into c-KIT high and low. And we showed that the c-KIT low stem cells tended to have increased inflammatory signaling on RNA-seq. You can see increased TNF, IL-6, inflammatory signaling, and reduced OXPHOS and cell cycling markers compared with the c-Kit high LTHSC. And you could see that, and this is just in vitro treatment with TKI, and you can see that this is the c-Kit high and the c-Kit low, and that after treatment, you have this accumulation of c-Kit low LTHSCs, confirming or consistent with the finding, which we had from patients that this population may be more of a TKI-resistant stem cell population, and maybe the important population to study. So we've also looked at this by single cell analysis. And this is again from the mouse model. And these are patients, these are from mice that were either treated with vehicle or with TKI. And what we found was that, what you can see is that with TKI treatment, there's accumulation of cells on this side, which are the more primitive populations, which are the quiescent HSCs, some activated HSCs, and a reduction of the more sort of maturing and activated stem cells. And that if you look at the persistent versus the depleted population, that the persistent population showed increased levels of, had some reduced levels of certain metabolic, had a reduced metabolic profile, whereas they showed increased levels of inflammation of stem cells and quiescence-related genes. So this is all suggesting that, we can model some of the things that we're seeing in the patients within the mouse model. And one of the things that, came out from the study, from the gene expression study, was an evidence that there was increased expression of HIF target genes and the HIF regulome after TKI treatment. And we could also see that there was increased HIF expression, which was present both in hypoxia, but also in normoxia, suggesting that it was non-canonical enhancement of HIF. And we showed that HIF inhibition using economycin was effective in depleting the dormant LTHSC population. We did not see this population being inhibited with TKI, but we saw it being inhibited by HIF inhibition or in the combination of TKI and HIF inhibition. Whereas, with the and we also sort of showed that these were truly stem cells because we did secondary transplants to look at their regenerative capacity, and we could see that the HIF inhibitor treated stem cells from HIF inhibitor treated mice had lost ability to regenerate leukemia after secondary transplant. And then, if we treated the mice and then stopped treatment and followed the mice, that we found that there was improved survival and less recurrence of leukemia in the mice that had received TKI and HIF inhibitor. So this supported a role of HIF in maintaining the quiescent self-renewing stem cell. In terms of the mechanisms of this, we saw on single cell analysis, looking at the quiescent HSC population, that HIF inhibition was associated with increased oxidative phosphorylation activity. This was also seen, using a CNET assay, looking at metabolic dependencies, where we show that dormant LTHSCs develop more mitochondrial dependence after HIF inhibition, and that HIF inhibition led to increased ROS generation and led to increase in cell cycling. So this suggests that HIF inhibitor treatment leads to depletion of quiescent self-renewing stem cells, possibly by altering their metabolism and leading to stem cell activation. So, and this is, I think current studies, related to that are trying to understand, what are the mechanisms by which HIF gets activated, after TKI treatment, with the hope that we would be able to find ways to more selectively target HIF, to target these quiescent leukemia stem cells in future. The other part of this talk that I'm going to come to is related to the stem cell niche. And so the concept of the niche was, brought out by Dr. Schofield more than 50 years ago, and he defined the niche as a defined anatomical site where stem cells are sustained and reproduce, where differentiation is inhibited, but which have limited space, which also limits the number of stem cells. And people have identified a number of components of the stem cell, cellular components, secreted factors, inflammatory cells and cytokines, extracellular matrix, physical factors, hypoxia and metabolism, by which the niche influences the stem cell. So in the bone marrow microenvironment, the hematopoietic stem cell niche has been found to be associated with the vasculature, and especially with the mesenchymal stem cells that are associated with the bone marrow vasculature. There's also some role of endosteal cells in interacting with stem cells and their progeny. And then there are a variety of other cells within the bone marrow which also influence the hematopoietic stem cells and possibly the niche cells and contribute to niche regulation. So, so I think when we think of the other thing which we have seen and is that the leukemia itself alters the niche. And I'm going to show some data related to that to come. But the concept is that, there's the regular niche and you have a leukemic cell and you have normal cells in black. And, the leukemic cells have some growth advantage and they overcome the normal cells. But, I think what we are sort of starting to understand is that the leukemic stem cells are not really very sort of strong stem cells, and partly it might be because of this drive to divide and, and to generate leukemia, which is sort of works against, the ability to be quiescent and self-renew. And so in leukemia, we think, or in cancers in general, the cancer cells lead to a change in the microenvironment, which leads to reduced fitness for the normal hematopoietic stem cells, but which the leukemic stem cells are selectively adapted to, and that this may play a role in the development of cancer and in the persistence of cancer cells. And that's what we're going to just explore. So what we found was in our, and this has been shown in CML patients as well, but this is from the mouse model, that if we look at normal cells here on the left and CML mice here on the right, CML bone marrow showed increased expression of a variety of inflammatory cytokines. And we further showed that these abnormalities can persist after TKI treatment and during leukemia remission. And we also showed that there was increased competitive fitness of CML leukemia stem cells in CML bone marrow compared to normal hematopoietic stem cells. So if you transplant normal hematopoietic stem cells into CML marrow, they show reduced growth compared to normal hematopoietic stem cells in normal marrow. However, when you transplant CML hematopoietic stem cells to CML marrow, they grow just as well in CML marrow as they grow in normal marrow. So they are not inhibited by normal marrow. Getting back to what I was saying earlier about having an adaptive advantage. We looked at this further beyond cytokines to look at the mesenchymal populations within the marrow. We used flow to identify bone progenitors and stromal progenitors. And we found that these were both increased in their numbers in CML bone marrow. And that similarly to what I showed with the bone marrow transplants, if we co-cultured CML if we co-cultured CML LSCs with CML bone marrow, that they showed increased growth with CML mesenchymal cells, that they showed increased preservation on CML mesenchymal cells compared to normal mesenchymal cells. However, if we look at normal LTHSCs, they show actually reduced preservation on CML mesenchymal cells compared to normal mesenchymal cells. We showed that these changes in the stromal progenitors were related at least in part to TNF signaling, and that they showed an increased TNF signature. We did an interactome analysis which showed that the CML stromal cells showed increased expression of the chemokine CXCL1 and CXCL5, and the CML stem cells showed increased expression of the receptor CXCR2, and that TNF was playing a role in increased CXCL1 expression by the stromal cells, and that if we targeted CXCR2, that it had an effect on the leukemic cell, but particularly in combination with TKI led to depletion of the leukemic cells. This is both using an inhibitor as well as in a CXCR2 knockout mouse model. So we think what's happening here is that TNF secreted by the leukemic cells acts on these mesenchymal stem cells, these 63 positive cells, makes them secrete CXCL1. CXCL1 enhances proliferation and self-renewal of the leukemia stem cells, and that CXCR2 targeting on the leukemia stem cell can lead to stem cell depletion. And the current studies are sort of doing a broader assessment of the contribution of CXCL1 or its human counterpart IL-8 signaling through CXCR2, not only on the stem cells, but other CXCR2 expressing populations in the niche, which include neutrophils and the microvascular endothelium. So we'd also looked at this so the previous studies were done in vitro co-culture. So we also tried to target leukemia stems, the microenvironment in vivo. And the way we did this was using the CRE-LOX system. And we used different CREs, PRX CRE to target MSCs, and we tech CRE to target endothelial cells, and osteocalcin and ostrix CRE to target the endosteal cells. And we know that from previous studies, the CXL12 production from mesenchymal progenitors and to a lesser extent endothelial cells is required for normal HSC maintenance. And so we asked the same question in CML. And what we found was that if we disrupted the CXCL12, if you look at these PRX positive MSCs and stem cells, you could see that they sort of cluster together in CML bone marrow. But if you delete CXCL12, you kind of lose these MSC clusters, and you lose the clustering of stem cells with these MSCs. We showed that in normal hematopoiesis, depletion of CXCL12 from MSCs leads to depletion of stem cells. However, we were surprised to see that in CML, it actually led to an expansion of stem cells. But when we looked at this, we found that this expansion of stem cells was related to increased stem cell cycling. So, and then we showed that these cycling stem cells were still capable of, regenerating. These are, again, the same thing shown out here, that there are increased cycling stem cells, which can regenerate hematopoiesis in secondary transplant. But now, because these stem cells are cycling, they become sensitive to TKI. So whereas the control stem cells are not sensitive to TKI, the stem cells in the CXCL12 knockout mice are sensitive to TKI and are depleted, and they're depleted after secondary transplant, including depletion of donor LTHSCs. So the thought here is that the deletion of CXCL12 from the mesenchymal progenitors releases the CML leukemia stem cells from quiescence, allows them to self-renew, but also to cycle and thus become more TKI sensitive. So it's clearly that association with the mesenchymal progenitors is important for maintaining TKI resistance. We're still trying to figure out the mechanisms underlying it, but when we did RNA-seq of the stem cells from this wild type versus CXCL12 deleted mice, we found reduced expression of PRC2 target genes, suggesting increased PRC2 activity since it's an inhibitor. And we did indeed see increased EZH2 and increased sensitivity to EZH2. So I think one of the things that we feel is that when we release the cells from the microenvironment, we're allowing for increased PRC2 activity, which is potentially, and we're interested to understand what role this may be playing. We've some early studies in collaboration with Rulu, looking at this, shows that the H3K27 methylation sites, which are targets for EZH2, are increased in LSK cells from the CXCL12 knockout mice, as you would expect from what I just showed. And that we've looked at to try to see correlate these with gene expression. And it's interesting that you see a correlation with expression of targets, inflammatory transcription factors. So I think in the future, we are interested in understanding not only the mechanisms of niche-related regulation of PRC2, but also mechanisms by which PRC2 may regulate leukemia stem cell fate and TKI sensitivity, including interactions with inflammation. So, I think going forward, we have opportunities to analyze heterogeneity and tissue organization at the individual cell level, with the availability of advanced multiomics, imaging, spatial genomics, and, single cell metabolomics and things in the future. We hope to be able to use improved cell tracking methods, including cellular barcoding, use CRISPR-Cas and other genomic engineering methods for screens and genetic targeting, and also developed improved modeling, both in vitro with bone marrow organoids as well as with humanized mouse models. So just an example of, where this can take us. This is not our study. It's a study from a colleague, Tiong Ong, who's in Singapore, who did single cell analysis of patients at the time of diagnosis. And he divided the patients into three groups. These are some of the patients who would go on to have an optimal response. And these are the sort of patients who had a deep molecular response and were likely to be TFR candidates. And there were patients who had suboptimal response. They got some molecular response, but it wasn't deep. And then there were patients who failed treatment. And he did single cell analysis. And he was able to identify factors that were related to leukemia stem cells and related to the microenvironment, which correlated with what was going to happen to patients in the future. So he found that there was a sort of skewing towards this erythroid program in the leukemia stem cells that was associated with a better response, as well as NK cell abundance and a certain NK cell activity and function. Whereas, lack of that NK cell activity and, expression of inflammatory programs or MYC and other patient-specific features were associated with poor response. And, we would like to study this further, and especially, at the time of TKI discontinuation. And we were able to start doing this in collaboration with Dr. Kim in Seoul, South Korea, who actually has a collection of samples from CML patients collected at the time of stopping, and he kindly shared some of these with us. And this was a time we did bulk RNA-seq analysis in MNC as well as in sorted populations. And we looked at the expression of genes, comparing patients who subsequently recurred versus those who maintained TFR. And it was interesting when we looked at the gene expression that the prominent overexpression of different inflammatory chemokines and cytokines in the bone marrow cells from patients who recurred, as opposed to the patients that maintained response. And if you look at GSEA, you see the same thing. You see increased expression of inflammatory response in the mononuclear cells, as well as in different populations from patients who recurred, and altered reduced metabolic features in the patients who remained in TFR. So I think this is very interesting. We're currently in the process of actually analyzing single cell analysis from similar samples from TKI stop to be able to better define the cells and the factors which are associated with TFR versus recurrence at higher resolution, and use this to further guide us as to the mechanisms by which inflammatory signaling could potentially modify stem cell function and fitness following TKI treatment. So this is my last slide. And, just to say that there are a number of mechanisms that have been found. We think that the leukemia modified microenvironment provides an adaptive advantage to LSCs and regulates their sensitivities to TKI via inflammatory signaling, epigenetic modification, and metabolic adaptation. And we're hoping that, the work that we do in the next couple of years will give us better opportunities to target these mechanisms, to promote LSC attrition and leukemia generating activity, and to be able to offer TFR to more patients, with CML. But, I said, we're obviously always groping in the dark, and this is what we think we're seeing, but we're always, we'll have to figure out if what we see is true or not. And hopefully we're holding the right, the right part of the elephant. So I just want to thank all the people in the lab, current and past, as well as, collaborators at UAB and outside, with which this work was possible. I'm happy to take any questions.